Welcome, everyone. My name is Allison Jones, and on behalf of Canadian Nuclear Laboratories and our supporting partners, Atomic Energy of Canada Limited, Health Canada, and the Canadian Nuclear Safety Commission, thank you for tuning in with us today. CNL's Low Dose Research Webinar Series is an opportunity for us to help strengthen connection within the research community with a focus on lectures delivered by world-leading researchers pertaining to the effects of low-dose radiation from different sources. And we're certainly thrilled to have Susan Bailey with us today to present Twins and Telomeres in Space. Before I hand it over to CNL's Edward Azam to introduce Dr. Bailey, I would just like to draw your attention to the Q&A function at the top of your screen. Throughout the presentation, we encourage you to write in any questions you may have for Dr. Bailey. We will certainly look to get to as many of those questions as possible following the presentation. And with that, I'll pass it over to Dr. Edward Azam. Over to you.
Thank you, Alison, and warm greetings to everyone on this cold and snowy day in Ontario and in Colorado. Today is my distinct honor to introduce Susan Bailey, a pillar of the radiation research community, one who has made major contributions to the study of chromosomal damage induced by ionizing radiation, in particular changes that are induced during prolonged space travel in low Earth orbit and beyond. Susan is professor and radiation cancer biologist. She is a colleague to many in this audience. She has had a distinguished career in collaborative research at the Los Alamos National Laboratory and the Department of Environmental and Radiological Health Sciences at Colorado State University. Throughout her career, she has provided tireless service to the radiation research community, which earned her leadership roles. She is fellow of the Radiation Research Society and is past president. During the difficult times of the COVID pandemic, she has spearheaded key educational activities of the RRS, in particular the educational sunrise sessions during the annual meeting. She is chair and member of the program committees of numerous national and international meetings, including the upcoming International Congress on Radiation Research in Montreal, Canada in August of this year. Susan is mentor to numerous doctoral students and postdoctoral fellows. Her studies have been marked by innovative approaches in detecting DNA damage induced by different types of radiation in environmental and clinical settings. Together with her team of colleagues, she characterized track interactions in basic dose-response relationships. Her innovative approaches have been applied in radiation biodosimetry, genome editing, and the design of probes to detect genetic abnormalities associated with cancer with a high level of resolution. As you can imagine, these contributions have been facilitating the assessment of disease risk during occupational activities and in clinical settings. In particular, Susan has shown that telomere maintenance is not only a key indicator of general health, but is also a highly relevant biomarker of aging and age-related degenerative diseases. Her research is one of 10 investigations selected by NASA for the historic twin study, as NASA contemplates to return humans to the moon and plan to reach Mars, and is currently focusing its efforts to launch human space life science research into a new era of molecular-based studies. More recently, Susan and her team have been assessing changes in telomere length and telomerase activity in the space and Earth-bound twins, as well as in a cohort of other astronauts, with astronauts wanting to learn in depth about her work, as shown here with astronaut Shel Lindgren visiting her lab. Today, Susan will share with us findings and thoughts on twins and telomeres. Thank you, Susan. Thank you.
Oh, thank you so much, Ed, for that really very, very kind introduction. I very much appreciate that, and I appreciate the invitation as well to speak with this audience today, and also to share and highlight some of the studies that you just mentioned. I will definitely be reviewing some of the findings from our twin study. Can you see my presentation all right? Is everything good?
Yep, looks great.
Okay, thank you. I will be, like I said, highlighting some of our studies, our NASA studies, and really highlighting the potential mechanistic role of chronic exposure to space radiation as underlying the changes that we're seeing in telomere length dynamics, as well as the persistent DNA damage responses that we observed. Individual differences in response were certainly also observed and underscore the need for personalized approaches to evaluating human health effects and of long-term space exploration. There's little doubt that we are certainly living in a very exciting time for the space program, and Dr. Azam brought that up, that we really have witnessed the dawn of a new era of human space exploration. We have commercial rockets and vehicles like the SpaceX Falcon 9 and Crew Dragon launching NASA astronauts and even private citizens to the stars. We've also witnessed the first major step toward returning to the moon and lunar exploration with NASA's successful Artemis 1 mission late last year. Artemis 1 was an uncrewed moon orbiting test flight of their new SLS rocket and the reusable Orion spacecraft. As such exploration missions and even space tourism become realities, and more of us experience the extreme environment of space for longer and longer periods of time, it becomes more important than ever that we better understand the human health effects of long-duration spaceflight. The International Space Station, or the ISS, has supported a continued human presence in low Earth orbit, or LEO, for more than 20 years. And even life aboard the ISS, which travels at about 250 miles above the Earth at a speed of around 17,000 miles per hour, even that represents a very unique lifestyle associated with extraordinary challenges and stresses. It also affords us an amazing view and perspective of our own home planet. Here you see a picture taken by Scott Kelly of the Northern Lights, taken from the ISS. And it's absolutely one of my favorites. So with that, we'll go ahead and I'd like to start by introducing some of the stars of the story that I'm going to be telling today. Here you see, and thank them for participating in our study. Here you see in the front, we've already heard about Shel Lindgren, NASA astronaut Shel Lindgren, and then behind him is Japanese or JAXA astronaut Kimiya Yui. And over here on your right is a fellow you might recognize as Scott Kelly, who was selected as NASA's first 1-year mission astronaut and flew the 1-year mission from 2015 to 2016. And certainly lots and lots of PR and excitement around the 1-year mission, but most of the excitement was really around quite a different fact, and that was that he had a twin brother. I was also going to mention that you might have even heard some fake news about Scott Kelly, and that was that he returned as a space mutant, which of course was not true at all. And in fact, there was actually a lot of real science around the 1-year mission for a whole year before the flight, as well as a year during, and then a year after. Many samples and many studies were performed. But as I mentioned, I mean, it was really during one of the early press conferences with Scott Kelly that he brought up with, is NASA doing anything? Because I have an identical twin brother, Mark Kelly, who was a former astronaut as well as a former Navy test pilot. And so it didn't take very long to realize that, the perfect experiment was sitting right in front of them, and that is that they had 2 individuals that were very similar in nature and nurture, and that is really how the twin study was conceived or born. The twin study was the first time that NASA really ventured into this whole area of omics. So looking at DNA and RNA, genomics, transcriptomics, proteomics, metabolomics, and all of them that you can imagine. But it was a very comprehensive study, and it was really the first time for many of these different studies. And so I'm going to just briefly highlight for you the 10 different studies from around the country that were selected for the twin study. And when you think about the fact that, some of these molecular studies require some just very basic techniques that we take for granted here on the Earth, but even something like pipetting in microgravity is very difficult. Tips flying all over the place, and liquids, how do you manage all that stuff? So here we see Dr. Feinberg and his team from Johns Hopkins, and they were actually involved in getting some of this worked out and getting some of the just even basic pipetting worked out. And they actually paved the way for the first DNA sequencing that was performed in space, which was done by Kate Rubins, as you see here, in 2016. And sequencing is now done on the space station fairly routinely, or is at least routinely as anything is in space. This is Dr. Chris Mason at Weill Cornell, and he and his team were also very interested in looking at DNA and RNA methylation and gene expression patterns. So again, they were very involved in this, and showed that in fact most everything, although there were certainly changes in gene expression during spaceflight, most of the values returned to pre-flight values, really demonstrating the amazing adaptability of humans to spaceflight. Scott Smith and his team at NASA did the biochemical profiles, so again profiling blood and urine collections before, during, and after spaceflight. And it's certainly true that, we are what we eat, and diet, of course, is a very important part of life. Here you see Scott Kelly and Kjell Lindgren eating the first space salad, and just from lettuce that was actually grown on the ISS. And certainly that is going to be a very important thing as we move forward, to be able to grow some of our own food. I'll also mention that, the diets, of course, on station are very defined. They can be very healthy, but they can also be very boring. And astronauts do complain some about that. Scott Kelly lost about 15 pounds while he was during the 1-year mission. And part of it is diet, but part is also that they do a lot of exercise to combat the muscle wasting and bone loss that they experience during in microgravity. Dr. Mignot at Stanford was involved in really the very first test of a vaccine in space. So you see on the bottom there, Scott Kelly and Mark Kelly each giving themselves a flu vaccine. And in fact, the immune response was very similar, whether in space or on the ground. So that was a very important finding. Stuart Lee and his team at NASA were involved in doing metabolomic studies and looking really at cardiovascular effects, monitoring artery structure and function during spaceflight. You can see some of the equipment here that they have on station that they use to do that with. And again, they found as well that most of the values return to baseline within about 6 to 9 months post return to Earth. Here you see Brenda Rana, who is at UC San Diego, and she and her team were involved in doing proteomic studies, particularly interested in looking at fluid shifts. So fluid shifts are experienced once you get into microgravity, and instead of fluid, staying more around your legs, it actually goes more up into your head, and astronauts do experience increased pressure in the brain and the eyes. It can affect their vision and blood pressure. And you can see here Scott Kelly and Kimia Ewing again using some of the equipment on station to measure some of these pressures. Matthias Basner at University of Pennsylvania was studying cognitive performance during spaceflight using a whole battery of computer-based tests. And not only the twins, but most of the astronauts actually do go through this type of testing. And what they found with the twins was that, in fact, Scott Kelly did suffer some decline in cognitive function post-flight, so once he came back to Earth. And it took him about 6 to 9 months to really recover. And even in his book, his autobiography, Endurance, he talks about how it was 8 or 9 months until he really did feel like himself again. And if you've ever wondered what the outhouse on the International Space Station looks like, this is it. It kind of reminds me of a 2001 Space Odyssey, that it's going to require some instructions for sure. But this was a very important study done by Fred Turek and his team at Northwestern, in that it was the first time that anyone had ever analyzed or looked at the microbiome during spaceflight. And as expected, it changed during spaceflight, but then it also changed fairly rapidly when they got back to Earth and returned to normal. So I hope that whirlwind tour through the twin study helped you appreciate the comprehensive nature of the twin study. And it was also at the very forefront of this idea of personalized medicine for evaluating the health effects of spaceflight in individual astronauts. And there's no doubt that certainly Mark and Scott Kelly are the most profiled pair on or off the planet. And they're also some of the most interviewed. One of the questions, interestingly, that was often asked is whether or not Scott would return to Earth younger than his brother Mark. And this was often in the context of watching Interstellar or Einstein's twin paradox. But it's those that have done those calculations assure us that even if it would happen with the International Space Station, it would only be a few milliseconds. But the question of aging and aging-related diseases and space travel really still remains a concern, and that's because it could very much affect performance during a mission, or it could also be a long-term effect of long-duration spaceflight. So that's really where we come into the twin study, in that we were the first to evaluate a biological marker of aging in the astronauts. And what we proposed was that telomere length dynamics, or changes in telomere length over time, would provide a very informative biomarker of health, general health, of aging, and of disease risk. And the reason for that is that telomeres, the telomere length, really integrates all of the different exposures and unique stressors and experiences that astronauts undergo during spaceflight. And things like microgravity, for example, and space radiation exposures, the isolation and very limited diets all really contribute to changes in telomere length. And here you see Kjell Lindgren again. You saw him in the previous slide flying the Colorado flag in the cupola of the ISS. Here he's on a second 6-month mission. He just recently returned. For the second mission, he commanded the NASA SpaceX Crew 4 to the ISS, and now has accumulated 311 days in space. So he's getting very close to being in space as long as Scott Kelly has been. So I've already talked about telomeres and how important they are and why we think they're valuable biomarkers, but I haven't really told you what they are. Telomeres are just the natural ends of linear chromosomes that shorten with cell division, and thus with aging. So they can be really considered kind of a mitotic clock, and are in fact a hallmark and driver of aging in humans. What's also becoming very appreciated is that it's not just cell division that shortens telomeres, but things like inflammation and oxidative stress and a whole host of lifestyle factors like nutritional and psychological and physical stresses, as well as environmental exposures like UV and ionizing radiations, all contribute to telomere erosion. So it's the other very important idea is that it's not just a snapshot, but it's this idea of how quickly telomeres are shortening over time, or telomere length dynamics, that's really provides an informative biomarker of aging. I'll also just mention quickly that telomerase is the enzyme that can maintain telomere length, but it's really not sufficient in the majority of our cells to do so. So that's why telomeres shorten as we age. It's also true that telomeres can give us an idea of aging, but they can also be a biomarker of the associated risk that goes along with age-related degenerative pathologies. So a lot of the things that we associate with aging, which could include things like reduced immune function or loss of fertility, dementias, cardiovascular disease, and cancer, are also associated with shortening of telomeres. So telomere length dynamics then is this idea of changes in telomere length over time, and it really captures or integrates this interplay between genetics and life experiences and exposures. So telomeres can be viewed really as sentinels that provide this information and can help be predictive of health, aging, and disease risk. So we've talked a little bit too about space radiation, and certainly of interest to this audience, and it is most definitely one of the major hazards associated with long duration space flight. Even the ISS in low Earth orbit is protected somewhat by the Earth. Certainly we are on the surface of the Earth. We're protected from the more damaging types of radiation, and as is the ISS. But even aboard the ISS, they are exposed to increased levels of radiation. For example, 6 months on the space station is about, astronauts receive about 100 millisieverts exposure, and you can compare that to natural background or average natural background on the Earth of about 1 to 2 millisieverts per year. If we think about, well, what about shielding? Can shielding help with the problem? And of course, that's not exactly, easy to do. It's very limited because shielding is heavy, and sometimes it's even non-existent, like in spacewalks or EVAs. So here you see Scott Kelly out on a spacewalk, the picture taken by Kjell Lindgren, and you can see a reflection of the Earth if you look real carefully there in his visor. So again, the point just being that shielding is problematic and can even in some cases make things worse when you consider secondary particles like neutrons. So basically the real problem is that as we journey further and further away from the Earth, as we go to the moon and even on to Mars, the doses will be higher and the types of radiations that people are exposed to, or equipment and plants and everything else, will be more damaging. The estimated dose, for example, for a 3-year mission to Mars is closer to around 1 sievert or 1,000 millisieverts. So really, something that we need to consider as we move away from the Earth. And in fact, the only time that we have been completely outside of the Earth protection is during the Apollo missions. And this is a particularly illustrative slide. It was a film taped to the ankle of Neil Armstrong during his 8-day Apollo 11 mission. And you can see here a number of, radiation tracks of radiation exposure. You can see sparsely ionizing X-rays and gamma rays, the kind of more blobby signals that you see here, as well as protons. There are some proton tracks, and then some very densely ionizing or high LET HCE particles, which is like this train track going through the middle there. So again, just very illustrative of, even on just an 8-day mission, the kinds of exposures that astronauts are going to be exposed to as we go on to the moon and to Mars. So we really did hypothesize that all of these different stressors that I've highlighted and mentioned here, including space radiation exposure, would surely add up to hastening or accelerating telomere length loss for astronauts as they're staying longer and longer times in space. So that we, as Ed mentioned earlier as well, we were so fortunate to be selected for the twin study, but we also had a separate study of 10 unrelated astronauts and their matched age and sex matched controls, ground control subjects. So we were able to then, between these two cohorts, really make comparisons between the twins that controlled for a lot of the genetic variability, and we could identify spaceflight specific factors that influenced telomere length. And then we could make comparisons with our unrelated cohort and really get a better idea of the role of individual differences and susceptibilities. And the other very important part about this, and I've mentioned it a few times, is the longitudinal nature of the study. And that is that for us anyway, blood samples is what we required. And they were taken then before, during, and after spaceflight. So really a very simple experimental design. Here you see the actual collection points that we had for the twins, and was very, very similar for our 6-month astronauts as well. But it was much more challenging in execution. And a lot of that is because, the astronauts have very busy schedules with training, and they're all over the world with the things that they do. But the most difficult ones were the in-flight samples. And though that's because they were taken on station. So you see there, a couple of the astronauts actually, they'll help each other take the blood draws. And they would do this right before a Soyuz returned to Earth so that the blood could remain at ambient conditions and be transported back to Kazakhstan, which then flew back with the NASA astronauts to Johnson Space Center. And we had those samples within about 48 hours of collection on station, which was really quite a remarkable feat. And I'm going to just go ahead and show you some of the results, both from the twins, which you see here. So this was, once we got that blood, we isolated DNA and did quantitative PCR on them to measure telomere length. And here you see the results for the twins. And as we go through this, you'll see this HR in green. That is our ground twin. So Mark Kelly in green. And then you'll see TW, who is the space twin or Scott Kelly, and his data is in blue. So you see the different time points all averaged here for telomere length. So you can notice that Mark Kelly in green stayed pretty stable both before, during, and after spaceflight. The most surprising thing, of course, is that when you look at pre-flight, then mid-flight for Scott, his average telomere length actually was longer than it was pre-flight, and then it shortened back again post-flight, very similar to what he was before he started. So this was really a very striking and unexpected finding. We were anxious to see, what about in the other astronauts. So when we combine it all, we can look at our control cohort, which you see here. And as I mentioned, that's just a age and sex matched healthy ground control population. And then we see the pre-flight values for all 11 astronauts that we had. And then you see this elongation of telomeres mid-flight, and then a significant decrease in telomere length post-flight. So it was very important to be able to have additional crew members so we could confirm these results as well as start to see some real significant differences. So the general trend then was that astronauts had shorter telomeres than our healthy age-matched controls pre-flight, and then this increase during flight and post-flight overall significantly shorter telomeres after spaceflight than they had before. We also used a second independent measure of telomere length to validate the PCR. And so to do that, we used a cell-by-cell analysis, or this telofish, which is just a fluorescence in situ hybridization approach. And what you have here is just a complementary telomere probe with the fluorescent tag on it. And so the telomeres light up in red. And you can see that very clearly here on this human metaphase chromosome spread. And then telomere length is just measured by intensity of the signals. So the brighter the signal, the longer the telomeres. And what's important here is that it is cell by cell, and you're really looking at many thousands of individual telomeres. And when we did that, here you see the twins again, and we see the same trends essentially. Again, Scott Kelly's telomeres longer during spaceflight, but even more importantly, when we look at all of the astronauts together, again, we see the same trends that we saw with the PCR, and the telomere elongation during spaceflight certainly did hold up with this assay as well. Telomere fish also does another really important thing for us, and that is that it allows us to generate these telomere length distributions. So you can see here, going from left to right, that you're going short telomeres to longer telomeres as we go left to right. And then here just telomere count, so how many telomeres had that particular length. And so you get these curves or distributions from short telomeres to long, shortest in yellow, mid-range blue, and the longest in red. Across the top, we see our Earth twin, and you get relatively stable distribution all the way through the study. And on the bottom panel, you see Scott Kelly, or space twin, and you can appreciate the very different types of distributions, and really much more dramatic types of shifts in the number of short telomeres compared to the number of long telomeres. And certainly mid-flight, which is right here, you can appreciate that again, there's this shift toward more long telomeres during space flight. And then when he comes back, it shifts back, and he has many more short telomeres post-flight than he did before. So very, very informative. I would also just like to mention that we did a completely separate study with prostate cancer patients, 15 of them that were undergoing radiation therapy, or IMRT in particular. And the reason I show this is that we saw something very similar with telomere length. And again, this is in blood. These were just blood samples taken from these patients before radiation therapy. This is in vitro radiation with 4 gray, I'm sorry. And then this is right after therapy, and then this is 3 months post therapy. So again, we can see kind of this trend toward longer telomeres immediately after radiation therapy. So it really did provide an interesting and totally separate way to support a mechanistic role for radiation exposure in this changes in telomere length that we were observing. Now, we also evaluated and were interested in looking at chromosomal instability. And one of the reasons for that, of course, is that ionizing radiation is a very effective inducer of double-strand breaks. So it breaks the DNA, which means it breaks chromosomes. And when that happens, these ends can get back together incorrectly. So chromosome aberrations or rearrangements really are a very informative signature of exposure to ionizing radiation. And the way that we chose to look at this DNA damage response really was by directional genomic hybridization, or DGH, which is just a single-stranded approach to looking at chromosome rearrangements. And you can see here that by using this, we can look at translocations, which are rearrangements between chromosomes. At the same time, we can also look at inversions, which are rearrangements within a chromosome that just make that signal hop over to the other side. So translocations, the signal moves between different chromosomes. Inversions, it moves within the chromosome itself. And for the studies that I'll show you, this is what we used, a single color 3-chromosome DGH paint. I mentioned that we can now actually use a whole chromosome or a whole genome approach, and with 5 colors, so we can karyotype, we can identify all of the chromosomes, and we can also do the same, essentially the same thing. And you can very easily, this is a much better illustration of what an inversion on chromosome number 1, for example, looks like. So moving forward, this is the assay that we'll be using for this. Now, when we look at our chromosome analysis, and we can also look at telomeres at the same time, this is what we saw when we looked at the combined astronaut cohort. So down here, you can see that, in fact, this is again our same kind of thing where we're looking pre-flight, in-flight, and post-flight. There's a significant increase in inversions during spaceflight, which is exactly what we would expect. But you also notice that the levels remained elevated post-flight, so they were persistent, and this went out for 9 months to a year after their return from space. The other really striking thing that we saw that was very spaceflight specific were these satellite associations, and I'll show you what those look like in a moment, just the short arms of the chromosomes really associating with one another. And that was important because those have been reported before for occupational workers, hospital workers, for chronic low dose radiation exposures. So that it was very consistent with that. We also looked at a number of these sister chromatid exchanges, or SCEs, and saw some significant increases during spaceflight. And that would be consistent with exposure to more high LET types of radiations. And then as far as the telomeres go, we saw a very interesting phenotype where the telomeres on the 2 different sisters were very different lengths, and they were just heterogeneous sister telomeres. And here's images of exactly what that looks like. Here you can appreciate really very easily how unequal or heterogeneous the telomeres on the 2 sister chromatids are. And what's important about that is that that is a hallmark of ALT, which is an alternative lengthening of telomeres pathway that does not require telomerase. So that could be some of, again, provide us a mechanistic clue as to what might be going on with telomeres. And ALT has been shown to be activated by high LET exposures as well as chronic oxidative stress. So this is we're very interested in this pathway and are continuing our investigation into that. And I mentioned the satellite association. That's what this looks like, the telomeres of those short arms of those chromosomes associating. And again, as I mentioned, that's an important biomarker of chronic low-dose ionizing radiation exposure. So just confirms a nice confirmation of the radiation exposure. I also just like to mention that it was interesting that we also found and just noticed that there was a very consistent decrease in white blood cell counts post-flight. What you see across the top here is the astronaut data, and over here you see with increasing dose, lifetime dose, you see an increase in inversion frequency. So that very, very consistent. Although we didn't have a lot of crew members we had this information for, it certainly was true that with increasing dose, we saw increasing frequencies of inversions. Over on the next panel, you see here that in fact with increasing inversion frequency, we saw decreased white blood cell counts. So we could use our chromosome aberrations like inversions, and in the bottom panel we had dicentrics as well. You can use it as a proxy for dose. So increasing dose, we see this decrease in white blood cell counts. And on the bottom, this is the data from the prostate cancer patient cohort that I mentioned earlier. And really the same thing is true, is that if we look at either dicentrics in A or inversions in B, lymphocyte counts are lower with the higher doses. So that again is gives us some perhaps mechanistic insight, suggesting that what we might be looking at is actually changing cell populations rather than telomeres themselves actually getting longer. Perhaps what we're looking at is a different cell population, like more stem-like cells, for example, that just inherently have longer telomeres. So we're very interested in following up on some of this in our future studies as we move forward. So I'll just go ahead and summarize where we are to date anyway, as using telomeres as these sentinels or biomarkers of space flight. And so for the twin studies, certainly some important findings, and starting with the fact that both twins started the study with relatively similar telomere lengths, which is very consistent with a genetic component to telomere length. Astronauts in general tended to have shorter telomeres than our healthy age-matched controls before flight. And, again, that could very much be related to the fact of the stressful lifestyle that they lead and in preparing for space flight. Astronauts also had lower levels of telomerase activity than the controls. I did not show you that data for the sake of time, and also just because it was very difficult and in fact impossible to measure telomerase activity during spaceflight. It didn't survive the flight back to the lab. So, we really don't know what's going on, a direct measurement of telomerase activity in space. The most really striking thing was this observation of significantly longer telomeres during spaceflight, and it was very independent of mission duration. We saw it in the 1-year astronaut. We saw it in our 6-month astronauts, and I'll mention we've even seen it in the Inspiration 4 crew, a 3-day mission. So it happens very rapidly. And it also didn't matter how we measured telomere length. We did the qPCR, we did the telofish, and we also did telomere sequencing. Chris Mason's lab did that, and we found, again, looking at telomere length with sequencing, we saw the longer telomeres during spaceflight. And we also, in collaboration with another of the twins investigators, looked not only at blood, but also at urine and saw the same thing. Telomere length then shortened very rapidly upon return to Earth. For Scott Kelly, we had samples within 48 hours of his return, and telomere length was very, very much shortened, significantly shorter immediately post return to Earth. And then in general, the telomere length seemed to recover over the following months, but it's still stabilized in the majority of the crew members to shorter than pre-flight averages. And there were these very dramatic shifts in telomere length distribution. And that's really where we found, in fact, that they had many more short telomeres after spaceflight than they did before. And that's really where we believe that the long-term health and aging implications are, is because these shorter telomeres really could be associated with accelerated aging and perhaps increased risk of some of these age-related degenerative types of pathologies. We also have some mechanistic insight, interesting, much of it provided by our cytogenetic analysis, but much of it pointing to chronic exposure to the space radiation environment, this low dose rate exposure to radiation. We saw persistent DNA damage responses during spaceflight. Inversions and satellite associations were really very specific to spaceflight and provided signatures of space radiation exposure. Chronic oxidative stress and mitochondrial dysfunction during spaceflight were reported in the twin studies and in fact correlated with our telomere length results, and is very consistent with the notion that it's this ongoing damage to telomeres and replication stress that is causing the elongation that we're seeing, or actually ending up in cells dying, which I think is also another real possibility. And these things did correlate with telomere length dynamics and could be the reason why we're seeing perhaps this transient activation of alternative lengthening of telomeres. I'll just quickly mention too that we've seen something similar, even not only in the prostate cancer cohort I mentioned, but also in twin climbers of Mount Everest. We had the good fortune to get samples from both climbing and non-climbing twins, and it's a somewhat similar extreme environment. And in fact, they too, during the climb and immediately after, had longer telomeres than they did to start with. So it seems to be a very consistent observation. We're feeling like, you know, since we're looking in blood in the majority of these studies, that and lymphocytes are very radiosensitive, that you would anticipate increased cell killing, which would correspond to our reduced white blood cell counts after spaceflight that we observed. And perhaps this redistribution of leukocyte subsets or changing cell populations is really what we're looking at. And that has been reported before and observed before by Brian Crucian and the immunology lab there at JSC. So again, just all of this kind of adding up to what I've been trying to tell you and weaving the story that it's probably not telomere elongation per se, but a combination of contributing factors like chronic oxidative stress and telomere damage triggering this transient activation of ALT, and these changing cell populations over time, really changing the profile of telomere length that we're observing. So you may be asking yourselves, well, you know, why should we even care about all of this? And you know, it's true that we all care about aging and getting older and living our best lives, or improving our health span, this idea of maintaining health span, not just lifespan. And certainly, although Scott Kelly came back from a year in space, briefly taller and 15 pounds lighter, and maybe even a few milliseconds younger, going to space is likely not a fountain of youth. And I think that's connected certainly to the fact that the long-term implications of having many more short telomeres than you started probably has a lot more to do with your long-term health consequences. However, I mean, it's also, if you think about aging in a larger context and kind of expand your vision, space can be viewed as really the next frontier of aging studies and potential breakthroughs, because that's going to be essential for the success of future space exploration and colonization, especially as the number and diversity of space travelers and even tourists increase. There'll be a wider range of age and health status to consider. We've already witnessed a civilian childhood cancer survivor in her 20s as the youngest to go to space as part of the Inspiration 4 crew. And we've even seen Captain Kirk or William Shatner as the oldest in space, our oldest astronaut at 90 years of age. So there's again, just a lot of excitement and a lot of things on the horizon. It's an exciting time to be a part of the space program. So I'll just end by thanking NASA for their funding of this research, and also the many different teams and people that were involved in generating the data that I showed today, my team here at CSU and a number of graduate students, and Lynn Taylor and Carrie George in particular, that really have been key in the assays and the work that we have done. But also Chris Mason's group at Weill Cornell has really been key in helping us integrate our telomere length results with some of these other findings, and getting at some mechanistic insight as well. And certainly I thank the twins investigators themselves. It's been certainly a pleasure and a privilege serving with them on these important studies. And I thank you for your attention as well. And I invite you all to stay tuned and buckle up, because we are going to the moon, and we may be there by the end of this decade. So with that, I'll just thank you all again, and be happy to take any questions.
Thank you very much, Susan. We do have some questions, and just a reminder, if we have time, still please encourage everyone to submit a question, and we'll do our best to get to it. So the first question, mechanistically, how do things outside of cell division, such as inflammation or radiation exposure, affect telomere length?
Yes, that's a very good question, and I think the best answer to that is that this idea of oxidative stress. So telomeres are very repetitive elements at the ends of our chromosomes, and they're very G-rich. So in humans, for example, the repeat sequence is TTA GGG. And there's 10 to 15 megabases of that at the end of our chromosomes. So there's a lot of G-rich sequence there that makes them very, very sensitive and susceptible to oxidative damage. And oxidative damage, I mean, that radiation does that too. It makes double strand breaks, but it also increases oxidative stress and damage. So I think that's what's going on, is that there is a lot of damage to telomeres, and they then experience replication stress. It can even, if you have enough of it, you're actually going to lose the end of your chromosome, and there's no way to get that back. So that really does contribute to telomere shortening, and why things like that really do seem to do it. But I do believe that it's primarily through increased oxidative stress.
Thank you. So the next question, and I believe you touched on this, but maybe clarify, how did the in-flight telomere length compare with the non-astronaut telomere length?
So for the twins, I guess we're talking about there, Scott and Mark. Yes. So Mark Kelly's telomeres, the ground control, really stayed very, very steady through the study. So, we took samples from him, as well as our other cohort of ground controls. We took samples from them at the very same time as we got the samples from the astronauts. And, overall, the ground controls stayed relatively stable through that whole time. So essentially close to 3 years for many of them, the before, during, and after. Telomere length remained fairly steady. And that would be consistent with the fact that most of these individuals, astronaut age currently anyway, the majority of them are between 35, 40 and 50, 55, that kind of age. And that midlife kind of range there is kind of a plateau of telomere length. You don't expect a lot of change during that time. So, and that was absolutely true of our controls, which is what made this telomere elongation that we're seeing in the astronauts, specifically during spaceflight, which is what made that so striking. It was just so completely different than the controls.
Okay, so the next question, did you use PNA fish for measuring telomere length?
Yes, so when I showed you the telomere fish, so the fish studies, we did use a PNA telomere probe for that. That's correct. So the sequence of the probe is simply, complementary to the TTA GGG sequence of telomeres, and it was on a PNA backbone. That is correct. And so then you just, do standard fish, and then you measure the fluorescence intensity of the signals. You look at it under the microscope, and we used image J and telometer. We've used a number of different things over the years, but that's how you go ahead and measure that fluorescence intensity as a as a proxy for telomere length.
Thank you. So how, well, can you talk to how the increased number of chromosomal aberrations and mutations contribute to risk of cancer? And then there's just a follow-up. Is the increase in chromosomal changes large enough to promote cancer?
Very good. Yes. So the answer to the first one is that, there's the chromosome aberrations are associated with many, many different cancers. I mean, that there's a very long and rich history of that. So we know that there are a lot of cancers that have chromosome aberrations, and they're causative. You disrupt, genes, you disrupt protein products, you get fusion genes and proteins that actually contribute to carcinogenesis. So chromosome aberrations, have a long history of being associated with cancer. The second part was, what was the second part? That does that increase the risk of getting cancer? Yeah.
Yes.
Okay, so yes, and in fact, yes, that it does. All you have to do is, disrupt the right the right gene, or get the right combination of things, and you can indeed increase risk. So that's why chromosome aberrations are used for that very thing. You can use them to identify a particular cancer, but you can also use them as an indication of increased risk if somebody has that gene. That's just that's pretty common. In this particular case, for what we did, I think the more important thing here was just that nobody's ever really looked at inversions, and part of that is because they're very difficult to detect. So we haven't really appreciated what role they might play in cancers, even though there have been a few cancers identified that were associated with a responsible inversion. So our studies were the first then to really show that, in fact, during spaceflight or radiation exposure, you do get these increases in inversions that persist. Another important aspect of that, because if a chromosome aberration kills a cell, you're not going to get a cancer. But if it's a chromosome aberration that persists and survives, and you've got, you have it in your cells, that is going to increase risk, and particularly if it involves something that might contribute to carcinogenesis. So we can't really say any more than it would just be expected that would increase risk for tumorigenesis. And that would be true of any chromosome aberrations.
Thank you. Okay, so I think we have time for a final question. What do you think about the fast contraction of telomeres following space flight? So from mid-flight high to post-flight low.
Yeah, that's a really good one too, because, I can kind of wrap my mind around the fact, on the other side of it, when the astronauts go up into space, the earliest time point we had there was at about 2 weeks for Scott Kelly, and telomere length was already elongated at that point. I can see how, so they've had, even 3 days with the Inspiration 4 crew. You're right off the bat, you're killing lymphocytes because of the radiation exposure. They're so radiosensitive, and so that I can understand. On the other side, coming back, there is a huge inflammatory response when they come back to Earth, and that's something they have seen over and over again. Again, I point to Brian Crucian's lab, as well as the other studies in the associated with the twin study and the inflammatory markers. But that's the only thing I can think of for on the return, that there's just this really heightened inflammatory response upon return, and that happens very, very quickly. And that would also be expected to really influence telomeres, and it must, because that's, we just saw, a very dramatic and quick shortening of telomeres. And so on that side of it, I would imagine that it's the inflammatory response that's doing that damage, and the result is that, really dramatically shortened telomeres. So I'll just say too that we are, at least we know now what questions to ask going forward. We didn't really know before. So we are part of the CIPHER team, which is just looking, again, doing many of the same types of studies on additional astronauts during all kinds of different duration missions. And we kind of know now a little more about what to ask. And so we are looking into some of the mechanistic underpinnings of some of these observations we've had. And I hope to have more of the answers for you next time.
Thanks, Susan. I'm just going to invite Edward to come back on screen.
Sure. Thank you. Thank you very much, Susan, for an amazing, amazing webinar. I know we have many, many more questions in there, and I hope, Alison, you can forward the questions to Susan, and Susan will recognize some of the individuals who asked the questions and can answer via email to them as well. Thank you, Susan, and thank you for the distinguished audience who have joined us today. We have been really fortunate to have you giving this webinar, Susan, and we look forward for all of us to tune in again in the very near future. Thank you, and have a wonderful day.
Thank you so much.
Day. Thank you.
It was a pleasure.
Thank you, Susan. Thank you.